Thank you to everyone in the UK for whom this is extremely early, but welcome to everyone across the world for whom hopefully this is a little bit easier. Hello, everyone. Welcome to our series of Alport Online workshops, the first in a while. Today, our workshop will focus on finding the missing variants in Alport syndrome. It's such a vital topic for patients who can sometimes get inconclusive genetic tests despite having the physical symptoms of the condition. Not having a conclusive test confirming Alports can cause massive mental health issues. And this is a topic that is not Alport specific, but relevant across other genetic diseases. A bit of a health warning too, some of the genetics content is quite technical. We've asked the speakers to explain the terms in as much detail as possible, but please pop your hand up or submit something to the Q&A if you're struggling to understand or you're not quite sure on a particular term.
So today we have presenters from Australia, Japan, China, and Europe, who are all going to talk about a variety of subjects in finding the missing variants of Alport syndrome. And apologies to those of you in the US, especially where the timing of this workshop is actually in the middle of the night. But it's amazing to see, as always, that we have a variety of international speakers with us.
I'm Patrick. I'm a patient and volunteer with Alport UK, and I'm hosting the workshop today along with Aura. Aura, do you want to introduce yourself a little bit?
Hello, everyone. I'm Aura. I'm a second-year medical student and a patient and volunteer with Alport UK.
So Archie Walker and Jane Parron are producing this webinar today, so do message them if you have any issues. Our scientists today will do short presentations, and then we'll have time for questions after each one of them. To ask a question today, please use the Q&A function. There's an option to remain anonymous, so you don't have to attach your name. We'll try and get through as many as we can today. Our scientific moderators, Julie Savage and Julia Hoffeler, will take those questions and put them to our speakers. If you're having any difficulties using any of the features today, hit us up using the chat function and an Alport volunteer will give you a hand. I'm just going to check the chat is indeed enabled. It doesn't look like it is. Anyway, chuck it in the Q&A, they'll be on it.
Chat, if you'd like to ask any questions of any of our panelists, please do put it in the Q&A function. But if you'd like to get in touch from a production perspective or any other issues, then please do message us. But if we can keep the questions to the Q&A function, it allows us to get through them all in a quickest way as possible.
Okay. To start our workshop, Aura is going to tell us about one of our Alport warriors, a woman from our closed Facebook community. Aura, over to you.
Thank you. So I'm going to briefly read the personal story. I'm in my 50s with a 25-year-old daughter and a 15-year-old son. My son and I were found to have a Col4 alpha-3 variant and an MYH9 variant. I have early cataracts, hearing loss, and high blood pressure. After genetic testing, I was referred to a nephrologist who didn't diagnose me with Alport syndrome, as my Col4 alpha-3 mutation is a variant of unknown significance. Because it's a variant of unknown significance, my son has not had his urine dipped or had a blood test for kidney function. My adult daughter sometimes has blood in her urine and is thinking of starting a family, but doesn't have a clear understanding of what this could mean without proper information. The hardest thing is the uncertainty and the unknown about the future.
I think this is a very common story for a lot of our patients, even the ones that are now diagnosed with Alport syndrome or have had a negative test in the other direction. It just helps sometimes to have a clear view, and that's not always possible with genetic testing, but hopefully we will learn a lot more about that today. There are loads of similar stories on Alport Warriors, our Facebook group, if you want to join. We hope the talks and discussion today will help shed some light on these challenging situations. We're always impressed as a charity by how much the genetics and kidney doctors do to help diagnose Alport syndrome, and we know it's a challenge in so many different circumstances. So thank you all for joining the discussion today. Right, let's carry on with some of the introductions to the people invited to share their experience of genetic testing and the complications of diagnosing Alport syndrome. Our speakers today are Dr. Bryony Thompson, a clinical scientist at the Royal Melbourne Hospital in Australia. Adam Bolzanos is a postdoctoral scientist at the Kids Neuroscience Centre and Children's Medical Research Institute at Westmead in Australia.
Tomoku Horinuchi as an associate professor in the Department of Pediatrics, Kobe University Graduate School of Medicine. Dr. Horinuchi is a pediatric nephrologist and clinician scientist who is also passionate about genetic kidney diseases such as Alport syndrome. Yangqin Zhang is an associate professor in the pediatric department of Peking University First Hospital in Beijing, China. Our expert chairs today are Professor Dr. Julia Hoffele, is a senior consultant at the Institute of Human Genetics at the Technical University of Munich in Germany. And our other chair today is Judy Savage. She's a professor at the University of Melbourne. Judy is an adult kidney doctor and one of the members of our workshop organising committee. And we thank Judy for having the idea for this workshop today.
It's fantastic to see some people who haven't presented, some people who have presented. I'm looking forward to all the talks today. That is enough from us, though. I think it's time we hand over to Judy. Would you mind sharing your screen and taking us away?
Okay, I was hoping I was sharing my screen already.
Not quite. It may be that we're spotlit.
So share away, Judy. You just have to reshare now.
Yep. Okay,
there we go. I think we can share your screen. We just need to make the presentation full. There we go. Perfect. Thank you, Judy.
Lovely.
Go for
Good.
it.
Thank you. And I'd like to thank the Alport Alliance for allowing us to have this webinar and hosting it for us. And I'd like to say hello to all of the patients and members of the Alport Alliance, as well as many colleagues who've worked on Alport syndrome for a long time. This is really a follow-up to the previous Alport collaborative meetings that we've held, and it was initiated because a couple of people emailed me and said, why weren't we still having meetings? So we thought that this was an important issue and wanted to get it out there and have some discussion about it. It's not a new area. We have made recommendations before. I'd like to firstly acknowledge everyone who has helped me in my work on Alport syndrome and in the presentation today, especially Mary Huang and my collaborators, the ophthalmologists, as well as Alport Australia, who've helped us financially with research grants, and the Alport UK group, and Susie in particular. So as Susie asked me to go over the outcomes of this meeting and what we had hoped to achieve, and I think we all know as clinicians that there may be no genetic results from testing for Alport syndrome, but it is important to let patients know that also, because they're often used to a very definite outcome after a test. We hope that laboratories might consider alternative strategies to detect more variants, and we hope that also we will share more of the pathogenic variants that we've found in our own laboratories, so that functional tests don't have to be repeated by different labs. And we think that the strategies that we're discussing today may be useful in detecting the missing variants in other genetic diseases too. So the previous recommendations that we published in Clinical JSON in 2022, we said there was no uniform approach where there was a strong clinical suspicion of Alport syndrome, but no variant identified. But there were a whole range of strategies that labs used in differing order. They usually routinely reviewed the quality of the sequencing and coverage where they didn't find a variant in an Alport gene. They would also, as much as possible, reevaluate the family history, any renal biopsy findings, and may go on to ask about hearing loss and ocular abnormalities too. They usually considered another genetic kidney disease, and they may have referred the patient for another technique to detect a different kind of variant that we know is more difficult to find in the Alport genes. And they may have referred the patient to a specialised service or research laboratory for further study. So I just want to go back to the beginning and talk about Alport syndrome so that we're all thinking about the same issues. This is a genetic kidney disease that's characterised by hematuria or blood in the urine, kidney failure in some people, hearing loss in some people, and ocular abnormalities sometimes. And the clinical features vary with different kinds of inheritance. There's an abnormal basement membrane in the affected tissues, in the kidney, in the ear, and the eye. And the abnormality in the basement membranes results from pathogenic variants or mutations in 3 genes that we know well that code for collagen 4 chains. And these chains bind together to form a molecule, and loss of any one of those chains results in the loss of the whole molecule, and substitution with a different molecule that's related but is more easily degraded and results in the abnormal basement membrane. And we know that that abnormal molecule and abnormal basement membrane results in loss of the overlying cells, which normally form a very tight filter and prevent the loss of protein into the urine. We also know that the very classical description of Alport syndrome is a bit limited, and that there are other clinical features that can occur too. And we recognised at the last meeting we held that kidney cysts occur more often in Alport syndrome than would occur otherwise. And there may be some soft tissue, tissue muscle abnormalities that form tumours. There can be also aneurysms or weakening of the blood vessel walls. And sometimes Alport syndrome is found together with another kidney disease too. And we know in particular it's found with IgA glomerulonephritis. So there's 4 different kinds of inheritance of Alport syndrome, and the classical Alport clinical features are found in X-linked and autosomal recessive disease. Autosomal dominant Alport syndrome is where there's a single pathogenic variant in one of these Alport genes, and a lot of these people only have blood in the urine. This is a very common condition occurring in about 1% of the population, and X-linked Alport syndrome occurs in about 1 in 2,000 of the population. But males with X-linked disease have a much more severe clinical picture than females, and they tend to develop kidney failure, hearing loss, and abnormalities in the eye. Autosomal recessive and digenic Alport syndrome are both quite rare. We've said for a while that genetic testing is better than kidney biopsy for Alport syndrome, and this is because there are lots of advantages of genetic testing. We find out the diagnosis for certain, but we also find the mode of inheritance. We can also predict some of the clinical course for the patient too, and we're also able to test other family members definitively. So genetic testing, we used to say, was very sensitive and very specific for Alport syndrome, but I think what we've found in the last few years is that the sensitivity or the ability to pick up somebody with Alport syndrome is less than 90%, and we're missing up to 30% of people who have either of the common forms of Alport syndrome, X-linked or autosomal recessive disease. So for the patients who don't have a pathogenic variant identified or have a VUS, a variant of uncertain significance, they have no diagnosis, they're generally not treated, and they're often lost to follow-up. Their diagnostic odyssey and their testing continue. For these individuals, the mode of inheritance and affected family members aren't identified, and affected family members may be mistakenly used as kidney donors. It's not only this very specific situation where somebody has no variant identified, but sometimes we have individuals who have one variant identified but have a much more severe clinical picture than we would expect. And they may actually have an undiagnosed second variant and have autosomal recessive or digenic disease, or they may have an additional hypomorphic or weaker variant found too, and they could have a greater risk of end-stage kidney failure. So it's not just the individuals who have no variant, but sometimes the individuals who have a single variant who are disadvantaged here. So the missing variants in Alport syndrome, they may be absent in up to 30% of some series, and other cohorts show 13%, 10% or more missing. And it's not just in Alport syndrome, but it's in other genetic kidney diseases, including autosomal dominant polycystic kidney disease too. So when we published these results, we compared the kinds of variants that had been found up until 2016 and subsequently a very small cohort in 2021. And what we showed was that the type of variants that we were detecting changed. We were picking up a lot more missense variants and fewer of the other kinds of variants like deletions, etc. Initially, we thought this was good news because the average age of kidney failure was changing, but somebody pointed out to us that this was also the time at which whole exome sequencing had been widely adopted in 2021. And maybe the change in the variants that we were detecting was related to the widespread use of whole exome sequencing. So the topics that we're going to talk about today, I'm going to go over some of the phenocopies, so some of the other diseases that can produce the same clinical features as Alport syndrome. I'm also going to talk about some of the areas of the gene that may be affected that we may be missing variants in. Bryony has recorded a talk about poor exome coverage and large deletions. And Adam and Tomiko are going to talk about non-canonical splicing variants. And Yan Quinn is going to talk about synonymous changes, and Julia about mosaicism. One of the topics that came up at the meeting in London was that with collagen type 1 and osteogenesis imperfecta, again, they were not detecting all of the pathogenic variants in their patients. And they found that some of the enzymes that were responsible for processing collagen 1 had mutations in them in their patients. So there's quite a number of genes can be affected and cause osteogenesis imperfecta rather than having a mutation or all the mutations in the collagen type 1 gene. But so far, no processing enzyme mutations have been demonstrated in Alport syndrome. So the clinical indications for genetic testing for Alport syndrome, genetic testing is still, I think, recommended rather than renal biopsy examination, despite the issues that we've found. But the commonest indication is persistent glomerular hematuria, persistent meaning greater than for 6 or 12 months. We know sometimes it's intermittent. We know sometimes it's macroscopic. But there can also be proteinuria. There can be impaired kidney function alone. There can be familial kidney disease. There can be abnormalities in the basement membrane, and the hearing loss and the eye abnormalities can occur too. And we know about the extended phenotype. So how well do these clinical features suggest Alport syndrome? The hematuria, the distinguishing feature really is that it's persistent and it has to be arising from the glomerulus. So that these red blood cells have an abnormal size, shape, and hemoglobin content. And this is best seen on phase contrast microscopy. Now, I know that phase contrast microscopy is not available to everyone. And I also know that if urine is left in the fridge overnight, the red blood cells may lyse, and so dipsticks are often better to pick up those lysed red blood cells and to be able to detect hematuria. So here's a photograph of a red blood cell working its way through the break in the basement membrane. These breaks are only transient, lasting for 10 minutes or so, and then they'll fuse back up again. We also know that about 10% of patients who have a thinned basement membrane have proteinuria only. And we know that the penetrance of hematuria is about 2/3, and it's less in men than in women. That means that men with a heterozygous mutation in Col-3 or Col-4 and autosomal dominant Alport syndrome are less likely to have hematuria. The hematuria and X-linked Alport syndrome, I think it's highly penetrant in men and in women. I just want to emphasise that autosomal dominant Alport syndrome is 20 times as common as X-linked disease. And we showed this by looking at how often pathogenic variants were found in NOMAD. So what I've done is quickly go through some of the other genetic causes of the common clinical features in Alport syndrome and see how often pathogenic variants were found in ClinVar in NOMAD. So this is not really a very accurate estimation of how often these diseases occur, but it does give us a very quick approach to the relative significance of these different diseases. And I've taken the genes which are reported in the Genomics England panel, and here Col-3A1, myosin 9 and LMX1 beta are all found, or Col-4A1 is found about this as often as Col-4A5, but MYH9 nephropathy and nail patella syndrome are much less common than autosomal dominant Alport syndrome. And similarly, when I've looked at the genes in the proteinuria panel, and I've looked mainly here at the autosomal dominant and X-linked genes, they are also much less common in general, except for Fabry disease. Fabry disease often has proteinuria, sometimes a little bit of proteinuria too. And the common diseases, common causes of nephrotic syndrome that occur, especially in children, the carrier states are much less common. In terms of looking at the common genetic causes of end-stage kidney failure, and this is a paper which is from Rosa Terra's group, and the commonest causes here are autosomal dominant polycystic kidney disease, the Alport genes, and autosomal dominant tubular cystic kidney disease. And these are relatively all of the same population frequency. Hearing loss, people often use hearing loss as one of the typical features of Alport syndrome. And here you can see the typical high tone sensory neural loss, but a mid-tone sensory neural loss can occur also. But when I went through OMIM and looked at the different panels for these kinds of kidney disease, you can see that a hearing loss has been reported in almost all of the panels, and it can be reasonably common also. The Fleck retinopathy, however, in our experience, is quite a good marker of Alport syndrome. This is the central retinopathy, but the peripheral retinopathy occurs in women with X-linked Alport syndrome, as well as males with X-linked Alport syndrome, and also in autosomal recessive disease. And this is just a peripheral retinal photograph taken on a retinal camera that's used for diabetes screening. And optical coherence tomography is also a good test to make a diagnosis of X-linked or autosomal recessive Alport syndrome. In terms of the genetic causes of abnormal basement membrane, there's actually very few other conditions that do result in the typical thinning, or this picture here, which is from a mouse, not from a human, of Alport syndrome with a lamellation. In terms of X-linked genetic kidney diseases, because sometimes we see a brother and a sister who are both affected, and the male has a much worse disease than the woman, and we think, well, it's an X-linked disease probably, and it could be Alport syndrome. And you can see here that the conditions Col-4A5, X-linked Alport syndrome, and Fabry disease are found in about the same number, about the same proportions. Dent disease, in my experience, is much more common than appeared from this strategy. So clinical features that strongly suggest Alport syndrome, I think any one of these may be sufficient evidence for further testing. So where are the missing variants in Alport syndrome? You can see here that this is from HGMD and their description of or their catalogue of pathogenic variants. So missense non-glycine variants are reasonably common as a proportion of all of the missense variants, and non-canonical splicing variants were 24% of all of the splicing variants too, and 11% of all the missense and nonsense variants were found in the carboxy terminus. I think we don't look carefully enough at the carboxy terminus. It's hard to work out whether a variant is pathogenic when it is here. We know that all of the cysteine substitutions are pathogenic, but this is a highly conserved region. A number of pathogenic variants have been reported in ClinVar, and interestingly, it's conserved in the ColFRA3, 4 and 5 genes, but it's also a double. So this region, the amino acids are repeated, and I've put them here end to end so that you can see that. Finally, hypomorphic variants. We find it hard to interpret some of these. Some of these are very common. The glycine 953 to valine occurs in 705 individuals, but a recent publication has suggested that it's benign. But on the other hand, the leucine 1474 proline substitution in ColFRA3 also occurs very commonly and is considered to be hypomorphic and pathogenic. The non-canonical splicing sites, we'll hear more about this later, but these are also common. So missense changes that alter the splicing sites, they result in milder disease, but the missense variants usually result in milder disease, but some are splicing variants that can result in more severe disease. And we'll hear a couple of presentations on this in a minute. And synonymous variants, we'll also hear a presentation on this. We know that there's 6 pathogenic synonymous ColFRA5 variants in Lovett. So in conclusion, pathogenic variants in ColFRA3, 4 and 5 are the commonest genetic cause of persistent glomerular hematuria and the second commonest genetic cause of kidney failure. And there may be some strategies that we'll hear about today that will help us to deal with these missing variants. For example, a multidisciplinary meeting identifying the clinical features that are strongly suggestive of Alport syndrome, other genetic kidney diseases being detected with a larger panel, knowing that whole exome sequencing misses some common types of pathogenic variants, and many of the intronic splicing variants have now been reported previously with evidence of pathogenicity. I think we'll also hear about some tools to assess pathogenicity, especially of these splicing variants, better computational tools and modelling. And we'll now have more understanding of the different reasons why we miss some of these variants. Thank you.
Thank you, Judy, for this very comprehensive and very interesting presentation. I think we have one question from the chat. Maybe you could answer that, because there was a question if it would make sense to repeat probably a genetic testing which was years ago and which was negative. Would it make sense to do that?
Julia, I should ask you that question. You're the person who runs a laboratory, a diagnostic laboratory. I'm the clinician. So could I ask you to repeat that or to answer that?
Of course, I can answer that. I would definitely recommend to do that, because in the past, the genetic testing was not complete and sometimes you missed variants. So it would definitely make sense to do it with a new technique and with some more information. Of course, normally we recommend to repeat genetic testing after 2 to 3 years if it's negative.
If the person previously had whole exome sequencing, would you go back and look at the sequencing again, knowing that there are, more and different genes, that it could be a phenocopy?
Definitely, yes. Yeah, we also recommend it for other patients which have a negative exome sequencing to perform reanalysis because, I mean, the literature is new again, and there are new genes and new diseases, so it definitely would make sense to repeat that.
Okay, good.
Okay.
Thank you. Now, I think I am introducing our next speaker, who is Dr. Bryony Thompson. Bryony has provided a recording, so I hope that our tech team have got that up. Bryony is going to talk about the coverage by whole exome sequencing and how it can be improved.
Hello, my name is Bryony Thompson. I am a clinical scientist in the Department of Pathology at Royal Melbourne Hospital. I'm sorry that I cannot be there live for this Finding the Missing Variants in Alport Syndrome webinar. I will be talking today about why sequence coverage may lead to these missing variants and considerations for finding these missing variants when it comes to sequencing coverage. So this was likely touched on in the first talk. There are different types of Alport syndrome causing variants. And so this table here shows the proportion of, or estimated proportion of the different types of sequence variants that cause Alports in the collagen genes. So you can see here they're mainly single nucleotide variants, which are substitutions of a single base pair. So your missense, which would be many of the glycine altering variants, some synonymous variants, and stop gain variants, nonsense variants. And then also small indels, which are usually a few base pair deletions or insertions that can be in the reading frame or out of frame and will produce premature termination codons. So these are typically detected by our usual sequencing techniques, which I will cover later in the talk. And that is potentially why the proportion is so high for these types of variants. Then we also have copy number variants, which are larger deletions and insertions or duplications, and they can be within a gene or include multiple genes. So this is just a little figure showing the different types of copy number variants that you can come across. So there's deletions, duplications, but there's also inversions, so swapping around of regions, and insertions of new sequence. So the techniques that are used to detect these disease-causing sequence variants can differ and have differed over historically. So typically labs would do Sanger sequencing to identify these pathogenic or variants or disease-causing variants, but this is quite a intensive and low throughput technique. Here I've got a little schematic showing the structure of genes in DNA, and so there are exons or coding sequences and intronic non-coding regions which are spliced out. And so in for Sanger sequencing, typically what would be done is sequencing of the single exons using special primers to target those regions. And so those would require a lot of intensive work to design those primers. So in the early, I think, 2000s, a while ago, next generation sequencing was developed. And so this type of sequencing can be whole genome. So a whole genome sequencing, I've used this apple as an analogy. It sequences everything, but then you can do more targeted sequencing. So you can do what we call exome sequencing, which is very common these days in laboratories, where you target these coding regions, so these exons, and it's quite a smaller amount of data to deal with. A lot of sequencing data, and you can see it's about 1% of the amount of information that you get from whole genome sequencing, or one slice of the apple. And so the exomes, and so the exome contains about 20 to 25,000 genes, but then from there you can even go more targeted and use targeted custom panels, which typically focus on 10 to 500 genes, so even smaller slices of the apple. And so next generation sequencing is typically now referring to a technique called massively parallel sequencing. And so I've just included here a schematic. Don't expect you to understand what goes on with massively parallel sequencing, but it's essentially using paired mates together, and there's a lot of computational biology behind it, and sequence those what we call reads, and they're aligned to a reference genome, and that's how we find these sequence variants, looking for differences in the sequence compared to a reference genome. And so this is another way of looking at the differences between genome, exome, and targeted panel sequencing, to give you an idea about that there's different levels of coverage based on these different techniques. So genome, because it is everything, has a lower but more uniform coverage, whereas with exome sequencing, we've got these probes designed to capture the coding or the exons in genes. So it's more increased coverage, but it's not as uniform as genome sequencing. And then the targeted panels, because you are so targeted on these specific genes, is a lot higher coverage, which is quite useful when it comes to detection of copy number variants, but has its drawbacks. So I'll be talking about those further into the talk. So in the context of Alport syndrome, historically sequencing of the 3 collagen 4 genes was done by Sanger sequencing. So that's a bit more, takes a bit more time and low throughput. But now with the advent of massively parallel sequencing, there are other laboratories that have used targeted panels. And so this is not specifically for Alport syndrome, but for renal disease genes, say in general, so that you can leverage that panel for other renal disease as well. But then the drawback of using targeted panels is that when there are new disease associations identified, the exome has that flexibility to include these newly discovered renal disease genes. And then the genome is the best, or you get the most data out of them, but so everything is included, but it's expensive, and at the moment it's expecting to come down in price, and it's starting to now, but it's also a lot of data to deal with. So there's a lot of computation that goes into the analysis of these massively parallel sequencing or next gen techniques. So some of the limitations of these exomes and targeted panels is that you typically do need additional analyses to detect copy number variants, which is not always implemented and can be difficult to validate, especially in the clinical arena. And also because you're focusing on coding exons, so you're missing the deep intronic variants and other known coding variants. They're not detected, but there are the development of exome captures, so that's what called those probes in the reagents that are used in this type of sequencing that are being decided to detect reported ClinVar. So ClinVar is a variant database, likely pathogenic and pathogenic variants. But I had a look at the collagen genes in ClinVar, and there's currently only 2 deep intronic variants in Col4A5. There are none in Col4A3 or Col4A4 currently, so this would certainly miss putative deep intronic variants currently with exome sequencing. And then other limitations of exome and targeted panels is that there can be gaps or regions of low coverage in the sequencing because it is not uniform as whole genome. And so what can cause these gaps is depends on the content of the composition of the sequence. So for example, high GC content is very difficult to sequence. And it also can be dependent on the exome capture that is used. But one bonus you can find when checking this quality data, and so you need to check this quality data when to know what you've missed, is that sometimes you can inadvertently pick up hemizygous or deletions in Col4A5 in males. I should prefix that with. So this is an example of where we've inadvertently picked up a deletion in a male with Alport syndrome by looking at our coverage data. So that leads us into the detection of copy number variants from exome data. So as you could see in that previous slide, there are differences in coverage produced by the copy number variants, and so that is how that type of that information is used to detect copy number variants in exome data. So duplications will expected to have higher read counts, whereas deletions would be reduced, and so this has to be compared to a lot of controls, which is a drawback of detecting copy number. You have to have a copy number variance in this type of data. You have to have large database to compare to, and there I don't expect you to read all of this table, but I just wanted to demonstrate there are a lot of algorithms out there that are used to detect copy number variants in exome data, and this is a list from a review done back in, a couple of years ago, and they use slightly different methods, and so because there can be tendency for false positive copy number variants in this type of data, it's recommended to use more than one algorithm to detect copy number variants. But they are improving quite a lot these days. So there are other copy number variant detection methods other than using the exome data that have been a bit higher validation. Sorry, I just got interrupted. There are different methods to for CNV detection, as well as using exome data or instead of, and these are chromosome microarrays, which clinicians may be familiar with, which are used quite a lot to detect chromosomal abnormalities. They do have low resolution when it comes to small copy number variants, and if you do use these, the typically they're lower density microarrays were used. So you want to use high density microarrays, example the GDA array, and that's a global diversity array. And they may not detect single exon deletions or duplications. And there's a more target approach, which is usually more specifically for 1 or 2 genes, which is multiplex ligation dependent probe amplification. And so this has custom designed probes to detect small chromosomal abnormalities. And so there's limitations with all of these. And there are other computational analyses that can be done on exome data to possibly detect missing variants. And so it's possible to miss mosaicism, which will be talked about in a talk later. And so in this, the bioinformatic pipeline is key. So the computational analysis is key. So typically, germline pipelines do not detect variants with low allele fractions, which are typical, that can be typical for mosaicism. So maybe less than approximately 20% allele fraction may not be detected. For example, the genome analysis toolkit, which is a common pipeline, GATK, the haplotype caller is the algorithm that's used for germline variant detection, so typically in blood, versus MUTEC2, which is used for somatic variant detection. So this MUTEC2 would be much better at detecting mosaics, whereas the haplotype caller would not detect them at all, even if they're present. So that's another consideration. So you can get a lot out of your data if you know the right computational methods to use to analyze it. So where to next? Well, whole genome sequencing would be the best tool we would have to find these missing variants, because you can get everything. So the deep intronic variants and the other non-coding variants, and also structural variants. And so the deep intronic splicing variants will be covered in talks after this. And then additional transcriptomic analysis would be highly beneficial, which is RNA sequencing to detect aberrant splicing or expression, and that will certainly be covered by others down the line. So thanks for listening. Good luck finding those missing variants.
Okay, great. Thank you to Bryony, who's on her way to the hospital ball. So I'm going to hand over now to Adam Benazos. We haven't got any time for questions, and Bryony isn't here, but Adam, if you could share your screen. So Adam works on RNA splicing.
Judy, sorry for interrupting. We have some open question. Maybe you can answer instead of Bryony, because otherwise it's too confusing if we hold on all these questions.
Okay.
Probably.
Maybe we could leave it till later. I just think we're getting a little bit behind with the time, if that's okay. And I'll have a look at the questions and see what I can think.
Okay.
Yeah. Thanks. Sorry.
All right, thank you for the opportunity to talk. I'm going to present a brief overview on RNA splicing, splice-like variants, and a couple of tools we use to determine how they are likely to alter splicing. Okay, so I don't have a lot of time, so I'll just get straight into it. If you look at any one of our genes, they're made up of exons and introns. They're coding and non-coding DNA. So exons are coding, introns are non-coding, and these regions must be spliced out and the exons spliced together in a very precise manner to transcribe your mRNA and translate that into a functional protein. So variants in non-coding regions are very difficult to interpret because we don't know exactly how they're going to disrupt this process and what the consequences will be for the encoded protein. So these are the consensus splice-like motifs. If you're looking at one intron here and exons flanking each side, there is the essential splice site that's made up of this GT donor, acceptor AG, and branch point A. Right? They're almost invariable sequences, but you can see the surrounding motifs are also somewhat conserved, and they help guide the spliceosome to cut at the exon-intron boundaries, splice your exons together, maintain your reading frame responsible for the amino acid sequence, and encode a functional protein. A variant in any one of these sequences can disrupt splicing. And it normally does that by either removing part of the transcript that should be there or including ectopic sequence that shouldn't be there. So for example, if you ablate a canonical splice site, it might result in exon skipping, including ectopic intronic sequence, or activating a cryptic splice site, or a variant anywhere in the intron or exon could potentially create a cryptic splice site. And these will have different outcomes for the protein. You might translate a truncated protein with a piece missing, or it might encode a premature termination codon and result in loss of function through nonsense mediated decay. So it's important to determine what the outcomes are for clinical interpretation. So our lab provides RNA testing for families affected by rare monogenic disorders, and we're trying to establish RNA testing pipelines that can be translated into the clinical labs and provide RNA testing for affected families. If we come back to the splice-like motifs, we know a variant in any of the essential splice site positions is virtually guaranteed to disrupt splicing, but there are other positions that are more conserved, such as the last nucleotide of the exon and the +5 position really preferred to be a G, but a variant anywhere from the third last nucleotide of the exon to +7 can ablate the donor splice site. And these splice sites are recruiting the spliceosome machinery. They're proteins with an RNA inside that base pairs with the splice sites, but there are a host of other RNA binding proteins, intronic and exonic splicing enhancers and silencers that influence and regulate the splicing reaction. The acceptor site is a bit more complicated. We have the essential splice site at -1 and -2. And these variants are responsible for almost 90% of the pathogenic clinical classifications at the acceptor end. And if we look at the remaining slice of the pie, 28% are -3 pyrimidine to purine substitutions. So there's a strong preference for a CROT, and changing the CROT to an A or a G is highly likely to disrupt splicing. There's the branch point motif. It has this T and A motif where the T and the A are highly conserved, and a variant at either of those two positions will disrupt splicing, but they only form a small portion because they're harder to interpret, or sequencing might not cover it. You can have multiple branch sites and so on. But I sort of wanted to highlight a class of variants that's possibly underappreciated. It's the next biggest portion of variants that are responsible for the pathogenic classifications. So these are AG creating variants. Between the branch site and the acceptor site, if you create a new AG dinucleotide, essentially a new essential acceptor splice site, that can also disrupt splicing. So if we take variants in ClinVar and Lovid that meet this description, you can see there is a high risk zone where all the pathogenic variants lie. It's 6 to 7 nucleotides downstream of the branch site and 5 to 6 nucleotides upstream of the branch site, where the majority of the pathogenic variants lie. There's also a lot of variants of uncertain significance and conflicting interpretations. So let's take this variant as an example. It's got nothing to do with Alports, but we've performed RNA testing on it. It's a -13 C to G. So it's here at this position. This C turns to a G, and look, this gene's on the opposite strand, so we're going right to left. It creates this AG dinucleotide. We've got branch point predictor and branch pointer, two different algorithms to predict branch sites. There's one at -17. It's probably a little bit too close to be used efficiently. But then there's one at -26 that fits right in the middle of the bell curve of where branch sites sit as far as distance to the annotated splice site. Splice AI is a deep learning algorithm used for splicing predictions. It doesn't predict that this new AG is used as a cryptic acceptor because it doesn't have a great polypyrimidine tract or appropriately distanced branch site, but it does predict that the acceptor site, the canonical acceptor site, is ablated. And Splice AI outputs many scores, and you have to take them all into consideration. Not only does it ablate the acceptor site, but it's predicted to ablate the donor site. So we interpret this as a prediction of exon skipping. Now, if we look at some RNA sequencing data for this same region in the gene, so this is called the sashimi plot. It's RNA sequencing data from in blood from a few controls, and I'm only showing splicing from exon 12, but the spliceosome is not perfect. It makes, for lack of a better word, mistakes, like a polymerase, here the most dominant outcome is by far canonical splicing from exon 12 to exon 13. But there are also these rare splice junctions, use of a cryptic acceptor in exon 13 skipping, and exon 13 and 14 skipping. So these are naturally occurring, individually rare events, but they occur commonly across samples. And we developed a tool called SpliceVault that ranks these common mistakes across 335,000 RNA sequencing samples across the GTEx and sequence read archive databases. And you can put in your variant, and it will give you this information at the appropriate splice junction. So here we can see canonical splicing is the dominant event. It occurs across 17,000 GTEx samples and 130,000 SRA samples. And then the next three most common things are those events we just saw in the sashimi plots, exon 13 and 14 skipping, exon 13 skipping, and use of the cryptic splice site. So they're individually rare, but they occur commonly across 2 to 3.5% of the samples. And what we found is when you ablate the canonical splice site, 92% of the time it will activate one or more of those top 4 ranked events, and 70% of the time it'll activate the top ranked event. So this is RT-PCR and RNA-seq from the patient, just to show that We did in fact see the top 3 ranked events enhanced, use of the cryptic splice site and exon skipping and double exon skipping. And here in the RNA sequencing data, there's the exon skipping and use of that cryptic splice site. So just to summarize, a variant almost anywhere in a gene can disrupt splicing, but there are other positions to look out for. The last nucleotide of the exon, +5, -3, and these AG exclusion zone variants. So we use a combination of SpliceVault and Splice AI to predict not only if, but how a variant might disrupt splicing. And I don't know if it's worth it just yet, but potentially we could derive empirically derived Splice AI thresholds for these 3 Col4A genes to predict splice disrupting variants in the future, much like the ClinGen SBI did for their genes of interest. But we would need enough variants across the whole splice site motifs to power this sort of analysis. So I'll leave it there, and I'll be happy to take any questions. Thank you.
Oh, thank you so much.
Can I ask a question? Adam, that's actually lovely, but very complicated, especially for a clinician. So can you use, are there any tools that will actually predict if a variant is affect splicing? You know, can you use Splice AI to say this is highly likely to affect splicing?
Yes, you can. You can. We use an in-house tool that's not published yet, so I didn't talk about it. But Splice AI is very, very good. But it would be interesting to see what the Splice AI scores were for variants that we do have RNA testing for, and what thresholds were appropriate to predict that.
And your work is highly specialised. You are a reference laboratory where people with different diseases or labs looking at different diseases send their samples to you. It's not as though every laboratory provides this service.
No.
So it's highly specialised, and it's really just to let people on this call know that this happens. And although it's complicated, there are people who are expert in it who can. And I'll hand over to Julia to introduce our next speaker.
I probably have one open question which I could ask now. I answered most of the questions using the answer button, so everybody should see them. But I think on one question from the audience, it's asked if we have a negative DNA result, can we offer RNA testing, and where can we do it, and how can we do it, or does it make sense or not at the moment? Probably you can answer that.
So our grant is for patients within Australia, but it's basically only in the research testing realm at the moment. There's not, I don't know of clinical labs that will just provide RNA testing for any case. There's a lot of basically guidelines to be established for testing and interpretation and how it fits in the larger picture, and that's what we're trying to do, but it's just not ready yet to just have it accessible to all families that have a putative splicing variant identified by genetic testing at the moment.
Okay, thank you so much.
Julia, can you
unmute
yourself, please?
Yeah. Julia, would you like to introduce our next speaker? Thank you.
Sorry, I turned off my micro. So the next speaker is Professor Tomoko Horinuchi from the Kobe University. He will present how to detect splicing variants.
Thank you for your kind introduction. Good afternoon, everyone. I'm Tomoko Horinuchi from Kobe University. First of all, I'd like to express my appreciation to all of you for giving me this opportunity. Today, I'd like to talk about splicing variants, one of the missing variants, and explore the hints for their detection. This slide shows today's topics on which I'll talk. First, let me provide a brief background explanation. As you know, Alport syndrome is a progressive hereditary kidney disease caused by abnormalities in the COL4, 4, or 5 genes. Although in recent years, advancements in genetic analysis technologies have led an increase in cases diagnosed with autosomal dominant Alport syndrome. The most common is X-linked Alport syndrome caused by abnormalities in the COL5 gene. COL5 gene is located on the X chromosome, leading to severe symptoms in males. I would like to provide explanations for each of the variants mentioned, as there were questions in the Q&A about them. And there are various types of gene variants. In particular, whether NC domain, C-terminal NC domain is conserved or not is very important in Alport syndrome. For example, in the case of missense variant is mentioned non-truncating. Only one amino acid is replaced, so subsequent amino acids are made unchanged, and the NC domain is preserved. On the other hand, in case of nonsense variant, so also mentioned truncating, so truncating contains the nonsense variant. And in case of nonsense variant, that is stop codon is created by a nucleotide substitution, and the production of further amino acid stops, and the NC domain is not preserved. So I'd like to return to the topics of splicing. Here is the linear survival curve for Japanese male X-linked Alport syndrome patients based on genotypes that we published in 2020. Patients with missense variant is represented by the orange line, and those with nonsense variants by the black line. And patients with splicing variants are represented by the green line. So this includes a total of 422 patients, indicating those with large deletions and including those with large deletions and small endo. Therefore, patients with splicing variants make up approximately 17% of the total. It is important to note that this represents the proportion of locally genetically diagnosed patients. So missing patients who have not been genetically diagnosed are not included in the data. So there might be a potentially larger number of patients with splicing variants, I think. Next, I would like to explain splicing. So splicing is a crucial process in the context of central dogma of molecular biology, which describes how genetic information is transferred from genome DNA to RNA and then to proteins. Splicing is the process where introns, the non-coding sequences, are removed from the pre-messenger RNA. Actually, the exon regions required for protein synthesis make up less than 1% of the entire genome DNA. Typically, routine genetic analysis involves analyzing only the sequences around these exons. In other words, introns are removed, so they don't need much attention. However, when intronic variants cause aberrant splicing, and they are considered pathogenic, and so disease causing. And the most reliable way to determine the presence of splicing abnormalities is through messenger RNA analysis. So in cases where pathogenic variants cannot be identified through standard genome DNA testing, analyzing messenger RNA can be another method to identify abnormal transcript, which is effective in searching for missing variants. However, I will explain later, performing messenger RNA analysis on patients, especially on kidney tissue, is by no means easy. As for splicing regulators, as Adam kindly presented, several factors are known. So it is possible to focus on genome DNA changes associated with these factors. Or when intronic variants are actually identified, we can predict the presence of splicing abnormalities through in silico analysis. Furthermore, we have established in vitro assay using meningine analysis to assess the presence of aberrant splicing. As explained so far, intronic variants can also be pathogenic if they affect splicing. However, messenger RNA analysis is sometimes difficult because messenger RNA is often unstable and difficult to obtain from patients in sufficient quantity and quality. In order to overcome this problem, we have established a system called meningine to evaluating splicing in vitro. Hybrid meningine constructs were created by inserting a test sequence consisting of exons and their flanking introns around the target variant between two exons, exon A and exon B. The hybrid meningines were transfected into HEC293 T cells. After 24 hours, total RNA was extracted from cells and conducted RT-PCR. Furthermore, with the use of site-directed mutagenesis, it's possible to create sequences, even those that have been reported. This slide shows the samples of the results of RT-PCR. So we can determine the splicing pattern in various variants by comparing it to the wild-type pattern. In this electrophoresis, the upper bands represented the canonical normal transcript, whereas lower bands reflected aberrant splicing pattern. In this case, exon 27 skipping. I would like to introduce our research that utilizes meningines to assess the pathogenicity of intron variants. We conducted a functional splicing assay with hybrid meningine for 7 intronic variants in COL4A5 gene, which located approximately -10 to -30 bases away from the exon. So 1 was found in our case, and 6 were reported cases shown on the human gene mutation database as pathogenic. So this is the results of the assay, and so the meningine assay showed exon skipping in 4 variants, number 1 and number 3, 5, and our case. So and for exon skipping and 10 base pair insertion by 1 variant, number 2, and no change by 1 variant, number 6. So for 1 variant, number 4, it was difficult to assess the splicing pattern by some reasons, and our assay did not work. So we showed that COL4A5 intronic variants, which are moderately apart from exon, can cause Alport syndrome. And this slide shows the results of in silico analysis for these variants. In 2019, when we published this paper, while changes in various splicing motifs could be identified, in silico analysis alone was insufficient to predict the splicing pattern. However, recently, analysis using splicing AI, Adam presented, has become possible. So Splice AI can predict the presence of aberrant splicing more precisely. So splicing prediction by in silico analysis has improved over the years. In addition, we have also used this meningine to analyze the impact of synonymous variants and missense variants at the last nucleotide of exons on splicing, and have reported our findings. So we believe that this meningine is a valuable system for assessing splicing. Next, I will explain NGS analysis for identifying intronic variants, genome DNA variants. I believe that genetic testing practices vary significantly by country. In Japan, while whole genome sequencing or whole exome sequencing or RNA sequencing using NGS is gaining popularity, it is currently more common to base genetic testing on NGS targeted panel test. So in Japan, we currently perform genetic analysis for patients clinically diagnosed as Alport syndrome using an NGS panel that includes exons of podocyte-related genes. However, there are cases where no pathogenic variants are identified. For such patients, messenger RNA analysis is one of the available options. However, as mentioned earlier, analyzing messenger RNA from patients' derived samples is often challenging. So, we created the NGS panel encompassing all COL45 exons and introns. In this study, we conducted an analysis in the COL45 intronic panel of 10 patients with clinically diagnosed with XRAS, but no pathogenic variants detected by NGS podocyte-related exonic panel analysis. Among 10 patients, 2 pathogenic variants and 5 likely pathogenic variants are detected. So as the next step, it is essential to evaluate the pathogenicity of these intronic variants. As Dr. Lamy shown that the assessment of detected variants is sometimes, in some cases, difficult, I think. So we are currently attempting to assess the pathogenicity of these variants using messenger RNA analysis, functional splicing assays with hybrid medicines, and insect analysis. Among these, I would like to focus on the deep intronic variant in intron 6. This variant leads cryptic exon in intron 6. Cryptic exon is typically not included in the mature messenger RNA transcript, but can be spliced into messenger RNA under certain conditions. Including the patients identified here, we have experienced 3 cases where different variants in deep intron 6 have resulted in the formation of same cryptic exon by different genome DNA variants. This electrophoresis indicates that transcript from urine-derived cells in patient 1, 2, and 3, in addition to the normal transcript in the lower end. Transcript containing the cryptic exon is also formed, as shown in upper band. The cryptic exon consists of 147 base pairs containing a stop codon within it, resulting in a truncating transcript. And there have been reports of cases where the same cryptic exon is formed due to different genome DNA variants. This table shows the characteristics of each case. They are actually clinically diagnosed as Alport syndrome. I think this cryptic exon in intron 6 could be considered a hotspot. And we are also exploring the therapeutic potential of exon skipping therapy for this cryptic exon. Although this cryptic exon contains stop codon, when we successfully induce cryptic exon skipping, the transcript becomes entirely normal. So the principle of this exon skipping therapy involves using antisense oligonucleotide to block the exonic splicing enhancer in cryptic exon and skipping it by preventing externalization. We have introduced ASO to the patient's urine-derived cells to induce skipping of the cryptic exon. And with the results confirmed shown in orange bars, ASO successfully led to the skipping of the cryptic exon. Furthermore, we also considered other candidates for inducing exon skipping, such as splicing-related proteins. So we introduced a specific splicing-related protein in an in vitro system, which led the skipping of the cryptic exon, as shown here and here. So the overexpression of SRP reduced the externalization of cryptic exons. So, and we believe that splicing is a promising therapeutic target. We are trying to develop treatments using SRP, which may have lower specificity than ASO and could potentially target a larger number of patients. Lastly, I'd like to share a report where exon skipping therapy was effective, although it may not be related to cases of splicing abnormalities. The concept of this exon skipping therapy is to make the severe phenotype closer to the milder phenotype by manipulating splicing. We experienced that male patients with extras caused by exon 27 skipping did not develop ESKD at the age of 46. Exon 27 is an exon consisting of 105 base pair multiple of 3 bases. In contrast, on the other hand, there are reports that under exon, the patient with nonsense variants in exon 27 has developed ESKD at the age of 23. We thought that this difference in prognosis might be due to the difference in whether the NC domain is preserved or not. The concept of exon skipping therapy is to convert truncating variants into non-truncating transcripts. For example, in patients with stop codon in exon B, if exon B is a multiple of 3 bases, skipping the exon results in a non-truncating transcript. This treatment involves binding ASO to the exonic splicing enhancer in exon B and skipping it by preventing externalization. ASO was administered to mice model with truncating variants. Wild-type mice, gray line did not show an increase of albuminuria, whereas the vehicle group, black line, showed an increase of albuminuria. In contrast, ASO, orange line, significantly reduced the increase of albuminuria. In pathological findings, vehicle group showed inflammatory cell infiltration, interstitial fibrosis, and glomerulosclerosis, whereas ASO-treated mice did not show cell infiltration nor interstitial fibrosis, and glomerulus showed intact appearance, almost intact appearance. In addition, ASO-treated mice also expressed alpha-5 protein globally. And surprisingly, ASO-treated mice achieved the perfect survival rate as wild type. So I would like to continue our research efforts to make exon skipping therapy using ASOs available for patients with various genotypes. In conclusion, intronic variants can also be pathogenic if they affect splicing. Minizine is a simple but powerful tool for evaluating splicing. NGS intronic panel analysis can detect deep intronic variants. We are now striving for the development of exon skipping therapy by manipulating splicing. I would like to express my gratitude to Professor Nozu and my colleagues. Thank you for your kind attention.
So thank you very much for this very impressive presentation, Tomoko. I've two questions, if that's okay, because I don't see any questions in the chat. You focused your project on the COL4A5 gene. What about the other both genes? Did you look in the introns of these genes also?
Also, we are checking the COL4A3 and 4, but the accuracy of clinical diagnosis is higher in, we think, X-linked Alport syndrome. So firstly, we try in COL4A5 gene, and then as a next step, we try to analyze COL4A3 or 4 genes.
Okay, and just one other question, short question. At least in Germany, we use the ASOS as an individual treating option. It's not covered by the health insurance. Would this be an option also for the Alport patients now, as a research treating option?
So you can, you means that the treatment option in Japan is covered, not strictly covered, but we can adopt the health insurance as for the RAS inhibitors or other kinds of renoprotective therapy. But so such as exon skipping therapy or is not for at this time in not human, and other disease specific treatment is not yet available in Japan.
Okay, thank you so much. I think then we can switch to the next presentation.
Yeah,
Okay.
we did have one more question that's gone into the chat, and it said, how is the ASO administered?
So for mouse or for cells? So
Yes.
both of them. As for mice, we inject so intravenously injected, and as for cells, urine derived cells, just transfection method. We used transfection methods.
Okay. Thank you for a wonderful presentation. So our next speaker is another recorded session from Professor Yanqing Zhang. So I wonder if our tech team could get this presentation up.
Hello, everyone. It's my great honor to have the opportunity to participate in this online workshop focusing on missing variants in Alport syndrome. My name is Yanqing Zhang from Peking University First Hospital, Beijing, China. Today, my topic is synonymous variants in Alport syndrome. Let's see a case first. A 25-year-old young man came to our department for genetic counseling. He presented with hematuria, proteinuria, and elevated serum creatinine and declined estimated GFR of 49.6 milliliter per minute. He had a long medical history. Hematuria and proteinuria was detected for the first time when he was 3 years old after catching a cold, and no attention was paid until he was 20 years old. Laboratory tests showed high serum uric acid of 600 micromole per liter and increased serum creatinine and increased proteinuria. He had no joint pain then. He was prescribed with drugs including angiotensin receptor blocker, allopurinol, and sodium bicarbonate. 3 years later, at the age of 23, he suffered from gout with pain in the right foot. Laboratory tests showed the serum uric acid was 652 micromole per liter and increased serum creatinine of 150 micromole per liter and proteinuria of 1.36 gram per 24 hours, indicating that the kidney damage progressed. So he underwent kidney biopsy. And pathological findings showed mild mesangial proliferative glomerular nephritis under light microscopy and segmental thickening and thinning and laminated GBM under electron microscopy. And further examination showed he had no ocular lesion or hearing loss. He had a positive family history of hematuria. His mother and maternal uncle, maternal aunt, and maternal grandfather all with hematuria. So he was diagnosed as Alport syndrome based on the clinical manifestations, kidney biopsy results, and positive family history. And in order to confirm the diagnosis and the inheritance, genetic testing was performed. Variants were detected in COFO-A3 gene. One is a missense variant in exon 50 inherited from his father, and the other one is a synonymous variant in exon 13 inherited from his mother. The synonymous variant was located at 765th nucleotide of the coding sequence of collagen 4 alpha 3 gene, and the amino acid at position 255th. We can see though the nucleotide changed from G to A, the amino acid didn't change. According to the ACMG criteria, the pathogenicity of these two variants in COFO-A3 in this case were both uncertain. So the patient was diagnosed as suspected autosomal recessive Alport syndrome. In order to provide evidence for diagnosis and genetic counseling for this patient, it is critical to answer the question, is this synonymous variant in COFO-A3 gene pathogenic? So we conducted an in vitro experiment. Firstly, 2 mini-gene constructs were built and provided by a bioscience company in China. One is the mutant type mini-gene, and the other is wild-type mini-gene construct. To build these mini-gene constructs, the genome sequence segment containing 1,180 base pair, including exon 12, exon 13, exon 14, and the introns were inserted into the mini-gene expression plasmid. Here we can see the variant was located at the last nucleotide of exon 13. Then many genes were confirmed by sequencing before transfection into HERC293 T cells, and here are the primers for sequencing. Next, cell culture and transfection. HERC293 T cells were cultured, and the wild-type and mutant mini-gene constructs were transfected into HERC293 T cells, according to the manufacturer's instructions. After 24 hours, cells were harvested, and RNA were extracted from the cells and was reverse transcribed into cDNA. Then PCR for amplification of the cDNA. And finally, the PCR products were separated by 2% agarose gel and analyzed by Sanger sequencing. Here are the results. By aqueous gel electrophoresis, the amplification product of the mutant type minigene transcription was shorter than the wild type minigene transcription. And after single sequencing, we can see the mutant type minigene transcription skipped exon 13, which has 78 base pair. And the wild type minigene transcription was normal. The results proved this synonymous variants in COL4A3 causing abnormal splicing. Then we reassessed the pathogenicity of the two variants in COL4A3 identified in this case based on ACMG criteria. The synonymous variant in COL4A3 is pathogenic, and the missense variant in COL4A3 in this case is likely pathogenic. So finally, the patient was diagnosed as autosomal recessive Alport syndrome, and genetic counseling was provided for him. This has been published. And our case is not the first one who had a synonymous variant in Alport genes. In 2016, Julia and her colleagues reported 3 novel synonymous variants detected in 3 patients with Alport syndrome and thin basement membrane nephropathy. In the first patient, the synonymous variant was in COL4A3 gene. And the other 2 synonymous variants were in COL4A5 gene. And in their study, the synonymous variants was about 1.4%, and they suggested that synonymous variants would be a rare cause of Alport syndrome. In 2020, Horinoch and his colleagues reported 3 X-linked Alport patients who detected with synonymous variants in COL4A5 gene. And these synonymous variants were in exon 15, exon 29, exon 42, and the amino acid position at the 292nd, 786, 1302nd amino acid of alpha-4, of type 4 collagen alpha-5 gene, respectively. And they did the experiment to evaluate the pathogenicity. The mRNA from the patient's urine and minigene constructs of these 3 synonymous variants were used. And we can see the first variants caused 17 base pair deletion, and the other 2 variants caused exon 29 skipping, exon 42 skipping, respectively. So their results provided evidence for that synonymous variants resulted in abnormal splicing, and minigene was a useful tool for investigating the pathogenicity of synonymous variants. And in 2022, also, Horinoch and his colleagues reported another 4 synonymous variants in 4 patients with suspected autosomal dominant Alport syndrome. And after analysis of mRNA from the patient and in vitro analysis using minigene constructs, finally, 2 of the 4 synonymous variants were proved to be pathogenic by causing abnormal splicing, and 1 causing exon 12 skipping, and the other one causing exon 14 skipping in COL4A4 gene. Here is a summary of the 12 synonymous variants in Alport patients has been reported. 9 of them were evaluated for pathogenicity by analyzing mRNA from patients or mini-gene constructs. 6 were proved to be pathogenic by causing abnormal splicing. The rate is about 67%. In summary, the first thing we should pay attention is don't forget synonymous variants. Synonymous variants in COF4A3, COF4A4, COF4A5 could be disease-causing, especially in patients with the following situations. First, patients diagnosed or suspected with Alport syndrome. However, no pathogenic variants were found after NGS. Second, for autosomal recessive Alport patients, only one heterozygous pathogenic variant in COF4A3 or COF4A4 was detected. Third, for patients with familial hematuria or thin basement membrane nephropathy with no pathogenic variants found after NGS. In these patients, please have a look at the sequencing data again. Don't forget synonymous variants. The other thing you can use urine mRNA analysis or mini-gene method to investigate the pathogenicity of synonymous variants, because synonymous variants in Alport genes could cause abnormal splicing. We thank all the patients and their families and the grant to support our study and our team. Thank you very much for your listening.
Thank you, Dr. Zhang. There aren't any questions in the chat, but can I just ask you, what about amplifying the alpha-5 messenger RNA from urine? How easy is that, or how difficult is it? What proportion of patients would you be able to do that from? And do you have any tricks? Do you do it within a few hours, analyze the urine straight away, or how do you do it?
You mean extract mRNA from urine?
I'm just wondering, I know some people have said it's difficult to do,
yes,
but do you have any tips on how to get it to do it successfully?
Yes, we have PhD students. The trick maybe is for female patients it's easier, but for boys, especially young males, it's difficult to collect enough cells from the urine. And always the patient should collect with the research, and they need to drink about 2,000 liter. Oh, sorry, 2 liter water. And then maybe stay with the research about 2 hours. Then they collect the urine, and maybe for some patients we failed to collect once, and then another time when they came to our department, we will collect again.
Okay. And what about using hair roots as an alternative?
We didn't try this in our lab. And I remember for some patients, if we consider it may be massage. Am I right? Well, maybe Julia will talk it later. For these patients, the mutation is different in maybe the percent of the mutation is different in different tissues. So we will do other tissues. Thank you.
Thank you.
I think there's also one open question from the beginning of our meeting, but it would fit very well, I think, to you. There's one patient, Kim, she has a COL4A5 variant, and also a VUS on COL4A4. And she has a son which inherited the COL4A5 variant, so he's affected. And she has a daughter with the COL4A4 variant, which is a VUS. So she asks, can we somehow differentiate, is my daughter also affected by Alport, or can we do any experiments to solve this problem? Is she a dominant affected, or is she just healthy? Could you make an answer to that question, please?
Okay, I try to answer this. In China, we also see similar cases. I think the person who asked this question may be a mother, a female. So she has no her clinical manifestation is milder, and her son is Alport patient, and because he inherited the COL4A5 gene mutation, and her daughter is inherited her COL4A4 variant. In our department, we will suggest the mother and the daughter take it easy, and but pay attention. You need to do urine tests every 3 or 6 months. Maybe you don't be so nervous. It's better.
Maybe we could also recommend to reevaluate this VUS in 1 or 2 years again. Maybe it changes, that it's a likely pathogenic variant, or that it goes the other way around, that it's likely benign. That might be helpful.
Can I ask, did the mother have both of the variants?
Yeah, and she's slightly or no affected.
Yeah. So it's possible to have a look at the daughter, just, several occasions. That might be what you were saying, and see if she has hematuria, if she only has one variant. Okay, thank you. So our next speaker is Julia, who's going to talk about mosaicism in Alport syndrome. Thanks, Julia.
Okay, thank you so much for the introduction. Can you see my presentation in the full modus?
Yes.
Perfectly. So I was asked to talk about mosaicism and its role in missing variants. It's just a small presentation because I think it's a small topic. Still, we have these new techniques on next-generation sequencing and so on. So I would like to give you just a short overview about mosaicism in general, and then its role in Alport syndrome. Then I have a small case report, which I published several years ago. It's old, but I think it's still interesting. And then I made a small conclusion. So what means mosaicism? It means that you have 2 or more genetically distinct cell populations within one individual. So you normally you have the full information in all cells, and they are all the same. But it can happen that during the conception or after the conception, a variant starts, and then you have it in some parts of your cells. So you have normal cells and cells with this variant in your body. Here's just on the right side a picture. If you have a germline mutation or germline variant, you have your parents, your father. Can you see my point? Or maybe I just change it. So you have the father and the mother. And for example, there's something in the sperm. They're just before the conception, you have the variant, then they find together, and all cells which go out of this first 2 cells have the variant, and so the variant is in all cells of the fetus. If you have mosaicism, then you also have your parents, and you have a normal sperm and a normal egg, and the defect or the variant just starts in 1 or 2 of the cell population after conception. So your variant is only in some parts of your body. Just here another picture to show how it works. Normally you have this cells in the beginning, everything's normal and your body is normal. Or you have, if you have the variant starts in the zygote, so you have it in all cells, so the whole body is affected. But then if it starts somewhere afterwards, then you can have just one cell somewhere, and this cell also grows, or you have several other cells affected, and depending on where the cell is, which organ it develops, then you have the different phenotypes. So it can be on the lower part of your body, or on the arms, or at the head, or just localized at the hand or the lower arm of your body. So completely different depending on the cell which is affected. What kind of types of variants we have in this mosaic forms, but also in the germline mutations? So we can divide into chromosomal variants, which means normally we have 46 chromosomes, and it could be more or less. So we have a numerical variation, or within one chromosome we have structural variation. So maybe the genetic information is just turned around, or we have a deletion, or we have a duplication. So something is missing or something is duplicated. So you can divide concerning a balanced form. So everything is there, but it's located somewhere else, or it's unbalanced. Then you have there's something missing or something duplicated. And the other way is that you have directly in the genes which are located on the chromosomes, that you have there a variation or a defect in there. This can also be structural, but it can also be that there's just one nucleotide affected, and we call them single nucleotide polymorphisms. If it's a nucleotide which we think does not lead to a disease, or it's a variation, then we think CNV that it leads to a disease. So it's disease causing. So for the Alport, so far we know the monogenic variations are the most common ones. So how about mosaicism in Alport syndrome? As I know, there are only limited data on mosaicism described in the literature, so only a few patients. And the clinical presentation is completely different depending on the location of the mosaicism and the grade. So how much percentage you have in your body. Is it just maybe 10%, or is it 50% of your body affected? So that's the reason why also the clinical phenotypes can be completely different. So you have mild phenotypes and also severe phenotypes. And or at least in the past, it was pretty difficult to detect the mosaicism and the grade of the mosaicism. In the past, we only had Sanger sequencing, and you could not really see if it was a low grade mosaicism, that you really can detect them. Now we have this next generation sequencing, which make it more easier, but also if you have a low grade, it still can be missed. So I would like to show this one case we had several years ago, and in the meantime, I think we did not detect another one, but maybe we missed it. So we had a boy of German origin, and he had a microscopic hematuria at the age of 1 year, and followed by proteinuria as an infant. So it fitted very well to the clinical diagnosis of Alport syndrome. So he developed a hearing impairment at the age of 14 years. A renal biopsy was not performed at that time, so the parents denied to perform it, but I think because of the classical phenotype, the diagnosis was safe. So what about the family history? The mother had microscopic hematuria since the age of 40 years, and she also developed hearing impairment, and a myopia, but she had a regular normal glomerular filtration rate, so normal kidney function when she was older. And there were no other family members affected. So at that time, next-generation sequencing was not available or was just started. So we performed Sanger sequencing in the boy for the Col4A5 gene, because this was the most common one. So we started with this gene, and we could detect a pathogenic variant at the border from the exon to the intron. So it's just the first intronic nucleotide. So it's called a splicite variant at this border or boundary. So you can see that here on the upper picture, you have a healthy control, and this is the region of interest. And if you look in this boy, you see instead of the G, which should be the normal one, you see the A here. So it's completely different. So then we performed a targeted Sanger sequencing of the mother, just to find out what happens in her at this position. And we could detect a mosaicism. As you can see here, this higher peak is the G, but you have this small peak of the A, which is this mosaic, and she inherited one cell, so one egg, with the A to her son, and that's the reason why he has this homozygous variant in his Col4A5 gene. So what did we do just on research purposes? We looked in the mother on the leukocytes with a deep sequencing technique at that time. So it just mentioned that next-generation sequencing just came out. And so we looked on her leukocytes, on the urine, on hair roots, on mouth cells. And we could detect a low grade, but it was a little different. So again, here you have the healthy control on the upper picture, and here you have the blood cells. So it's about 40% of her cells have the variant. And here in the urine cells, you only have 7%. And also in the hair, you have just a low grade, so about 7%, and also in the mouth. So what we could see here, it's different in this mother. It's more on the blood than the other tissues, but it's there, so it's safe that she has a mosaicism, and so she also has probably a mosaic on her egg cells, and she inherited that to her son. Okay. So that's also the explanation if we have this low grade of mosaicism, that the mother is more or less healthy. She only has this hearing impairment, but the kidney function was normal at that time, and she was older. She was 46, and I do not know if she has now a more worse kidney function, but I don't think so. So just to conclude, mosaicism, at least to my knowledge, is pretty rare, but you should of note if you don't find any other variant which could be causative, and you have definitely a clinical phenotype of Alport syndrome, you should look deeper with the new techniques which are now available. And to my knowledge, there are only single cases so far reported in the literature. In general, mosaicism will be calculated about the reason for the phenotype in about 16%. Judy mentioned for Alport, it might be up to 5%. I think it's pretty rare, but you should always think about that. If you have the clinical symptoms of Alport syndrome, go into deeper and check for that. In female, it's a problem because you also have the X inactivation, which could influence the clinical phenotype of the females. Normally, it was said that females are less affected because they have the X inactivation, but it can also be the other way around, probably that you have an unskewed X inactivation, or what makes it that you have that the gene which has the variant is more active than the other one, than the healthy gene, and then you probably could have a more severe phenotype. So thank you so much for your attention, and I think I'm open for questions.
Good. Any questions for Julia? If anybody would like to put them in the Q&A. So Julia, you've only seen one patient with mosaicism?
Exactly. At least we only detect one patient with mosaicism.
Is there any evidence that it's more common?
I think so too, but I think at least in the other laboratories, they only check for disease-causing variants which are known so far, and that's a problem. There's a bias, I think, in the detection, that it's still not in mind that this could be also a reason, and you have to go further into the detailed laboratory results to check if there's a mosaicism or not. Or, yeah. I think
so. Some of the tools that Bryony was referring to, where you can actually quantitate how much a variant is there, is that useful in detecting mosaicism? And can I also ask, is whole genome sequencing, is that better for detecting mosaicism?
Just to your first question, I think it doesn't, or you don't have any benefit if you know about the grade of the mosaicism, because you don't know how is it in the ears, how is it in the eyes, how is it in the kidney directly. So you only know it from the tissues you get easily, blood, urine, hairs, and so on. But you cannot, or you don't know how is it in the kidney located. And you don't make a kidney biopsy in these patients to know about the grade. Your second questions concerning would genome sequencing help to detect mosaicism? I think, or at least I think it might be not that helpful, we expect, because I think the coverage might be the same in the genome and the exome, and if the mosaicism is also seen in the exome, you don't have any benefit in the genome. Genome is only for structural variants and for intronic variants, and probably also for mitochondrial causes, so I don't think you have any benefit for that.
But RNA sequencing would be useful. Yeah. Okay, any other questions in the chat? Otherwise, I might just make some concluding remarks, and I'll ask Julia to make some.
We have had one question in the Q&A there.
Oh, okay.
Yeah, I just see that there's the question concerning is X innovation offered by labs, by diagnostic labs, I think. And I think, yes, you can do that, at least in Germany, it's offered, that you can do that. The question is if it really helps. At the moment, we only have limited data on X inactivation. There's one publication from the 90s, where they did not find any correlation to the phenotype and the X inactivation when they looked into the blood data. And I performed also one analysis where we checked kidney cells sorted out from urine, and we also could not see any correlations. So I'm not sure, even if you would like to check the X inactivation, I'm not sure if it helps you for the clinical treatment for the patients, because there might be other modifiers except the X inactivation, maybe other genetic modifiers which influence the phenotype in females. There's also one additional question. Do we have de novo mutations in mosaicism? There are no data so far. Probably yes, but most likely on the X chromosome. So I think the COL4A5 gene would be probably the most affected one. At least we see de novo variants in developmental delays or so quite a lot. So why should it not be in nephrological patients?
Okay, thank you, Julia. And I've just got a few comments here which are very general, but just that variants in Alport syndrome are more complex than considered previously. And I think we've had some wonderful information today from really the world leaders in this area. I think it's important to review the clinical features to make sure this patient does have Alport syndrome. We use the eye manifestations to do that because the retinopathy is quite specific. The peripheral retinopathy is quite specific. I think we've seen that there is a lot of information in the literature about these unusual variants, about splicing changes, about synonymous changes that are pathogenic. We are also hearing that there are improvements in the computational tools too, such as Splice AI, that are very helpful in helping to decide whether a variant is pathogenic or not. And in addition, we've heard about different techniques that can be used to confirm the pathogenicity with the looking at the urine for mRNA size and expression, and also with the mini-gene assays. But some of these techniques are complicated and do require a lot of expertise in a reference laboratory. So I think that, and I think we haven't really touched on whole genome sequencing and RNA sequencing very much for the diagnosis, but I know that some laboratories are using whole genome sequencing because of the ability to detect more variants initially. So I think it really has opened my eyes as to some explanations for my patients whom I'm sure have got Alport syndrome, but their genetic testing has come back negative. Julia, would you like to make some comments too?
I'm fine, thanks.
Okay, all right. Good.
I answered so many questions offline.
I apologise for the technical issues, and I would like to thank again the audience who stayed to the end. I'd like to thank the Alport Alliance for hosting us too, and I know that they will have further webinars in the future. And we hope that we will put together a publication for this, and we may ask people if they have any splicing changes that they haven't published, if they'd like to put them into, firstly to publish them, but secondly to put them into Loved, which we curate, or to ClinVar, which everyone has access to, because I think that to get some lists of these variants together would be very important. and there may be some learnings, as we were starting to hear with what Tomoko was saying. So thank you once again. Thank you to all of our speakers, and thank you to the people, to Aura and Patrick and Archie and the rest of the team at the Alport Alliance. Thank you.
Well, thank you also from my side. All the best and have a nice weekend.
Well, thank you very much, everyone. For those of you that are staying, if you have any more feedback or questions about the talk today, we've got the workshops at alportsyndromealliance.org email that should be pasted to you in chat right now. But as Judy and Julia said, thank you so much for watching. And thanks to Kidney Research UK for their sponsorship. They help fund our Zoom subscription and help with general funding at Alport UK too, so we're indebted to them for that. If you do want to keep up with future opportunities for workshops, follow us on social media. On the Alport UK Twitter page, that's at Alport UK, and the Alport Warriors Facebook group. For those of you that would like a recording of the talk, we'll be popping it on our YouTube and Facebook sometime next week. Okay, thanks everyone.
Okay, thank you. And thank you to all the speakers. I have learned a lot.